Or no?
Hello, thank you for stopping here today. I would like to discuss the cell membranes we have got going on. So for my concept map, I've decided to have plasma membrane as the main point of topic, and we will be talking about proteins, channels, solutions that come from it, the ways in and out of cells through the channels, the proteins made of the plasma membrane, and then also made of, we've got our phospholipid bilayer, which are two layered arrangements of phosphate and lipid molecules forming cell membranes. Up here we have vesicles, and they're within or outside of the cell. They can be in a liquid form enclosed by lipid bilayers. And almost forgot about our scientists who created the models that help us determine that help us figure out how the plasma membrane works. I'm trying to figure out set it up right. Dawson and Daniele, they've got the sandwich model, which basically just is describing the plasma membrane as a sandwich squished together with the phospholipid bilayers, as well as the fluid mosaic model, which is more a representation of the cell membrane function. And we'll move on to page number 2, as I have so beautifully prepared for you. Going on to the phospholipid bilayer, remember it is right over here. And now we'll go phospholipid bilayer. What a beautiful transition. And here we have the hydrophilic head and the hydrophobic tail. The hydrophilic head, of course, loves water, as opposed to the tail, which is a water hater. And the hydrophobic tail of the phospholipid is composed of glycerol and phosphate molecules. The hydrophilic head of a phospholipid is made up of fatty acid chains. Talking more about the phospholipid head, we'll go into saturated fats and unsaturated fats, so the two different types. Of course, your saturated fats you'll get from meat, and they're in a solid form. The unsaturated fats are in a liquid form, such as oils. Jumping down to amphipathic molecule, it's so cool because it's both in the hydrophobic and hydrophilic head. It can be both. Now moving up to the detergent molecules, they are various membrane functions on how the membrane proteins work with them, and the fact that they denature them. They're polar charged and hydrophilic, so that's why it's going off of this tail here. And not such a beautiful transition, but this is where we are in the time of the night, going from detergent molecules as they play a role in the membrane proteins. We will just go into this messy page of proteins, and I will just read to you. We have a couple different membrane proteins. We've got our lipid-linked membrane, which is the association protein conventionally attached to lipids. You have your monolayer associate membrane protein, which interacts with the inner layer of the lipid bilayer. I'm going to kind of skip around a little bit just to focus on the membrane proteins, so read along. The membrane protein is attached to the membrane of the cell. The transporter membrane protein moves ions, small molecules, and proteins across the membrane. Then you've got your integral membrane protein. They're permeattached. You have your anchor membrane protein, which is binding the cell to other structures. Your receptor membrane protein transmits information based on the application of ligands. Then you have your enzyme membrane protein, which catalyzes the chemical reaction. I'm really tired. The two main ones would be the integral membrane protein and the peripheral, because the peripheral is like a temporary attachment and the integral is more of a permanent attachment. And there's really a lot going on with proteins like all the time. They do make up a major part of the plasma membrane. And once again, sorry, it's just that time of night. But we do have all of the definitions, all of the words on this video, so I'm just gonna read them and how they go off of each other, off of the proteins. Hopefully I already did the channels. Simple diffusion does not require help from membrane proteins, just like it moves from higher concentration to lower concentration. And the passive transport moves from higher concentration to lower concentration until evenly spaced out particles. Until they're evenly spaced out, they'll continue to move. Active transport energy required from low concentration until it moves over to higher concentration. And let's see, what did I miss? Oh, hi. All right, so we've got our channels and solutions. The channels, meditated passive transport, spontaneous passage of molecule. Then you've got your gated, I wish that this was like, okay. Your gated ion channels, which are excitable cells. Your ion channels, which are the pathway for charged ions through an area that would otherwise be impermeable. And your membrane channel, which is the passive movement of ions passing down to the electrochemical gradient. Wow, how cool. And the electrochemical gradient creates a free energy to perform work. Solutions. We have our hypertonic. I feel like I pretty much memorized this anyways. Okay, so we have our hypertonic solution, which is, one day I will figure this out. Okay. The hypertonic solution, which is the lower water concentration of a solute compared to the cell. The hypertonic solution, which is the shrink or swell capacity of a cell due to the external solution with a high solute. And the isonic solution, which is the same solute as the water concentration. Okay, so there are all my words on all of my maps. Sorry that it is not as beautiful as the concept maps I've made before. However, I do feel like I was able to execute the description and understand why they were related to each other. Alrighty, signing off. Goodbye.